George Wilding. Dr. George Wilding obtuvo su título de médico de la Facultad de Medicina de la Universidad de Massachusetts, donde también completó su internado y residencia en medicina interna. La investigación del Dr. Wilding se centra en el papel del estrés oxidativo en la carcinogénesis de la próstata y la progresión del cáncer. El Dr. Wilding ha publicado más de 235 artículos. También tiene una pasión y un compromiso con la educación, como lo reflejan sus más de 20 años como investigador principal. Actualmente tiene el papel de Vice Provost de Investigación Clínica e Interdisciplinaria del Centro de Cáncer MD Anderson. Les pido un gran aplauso para el Dr. George Wilding.
It's exciting to be here. Pleasure to be back in Puebla. Earthquake drills and volcanoes and everything else. But it's really a pleasure to see all of the enthusiastic young people interested in research. It gives you confidence that the future science is in good hands, in all of your hands. So I thank you for your interest in research. I thank the leaders of INCAN, especially Luis Herrera, for putting this together and inviting me back for another year. So we're going to be talking about prostate cancer today. And specifically, I'm going to focus on cellular metabolism. Not only cellular metabolism as it might affect the progression of prostate cancer towards castrate-resistant disease, but how there may be spots in cellular metabolism that would serve as good targets for therapy, and whether there are agents that we might be able to take advantage of in treating castrate-resistant prostate cancer. So I have no financial disclosures. And in prostate cancer, as many of you are aware, in the United States, there are just less than 200,000 men that are diagnosed with prostate cancer each year, and about 26,000 men die of prostate cancer. Now, fortunately, the majority of men are diagnosed with localized disease that can be effectively treated, cured, with surgery or radiation. And even men with lower grade disease, more and more are choosing for active surveillance or watchful waiting for their disease. But unfortunately, a number of men go on to progress and have disease spread to other parts of the body and develop metastatic disease. These men initially respond at a high rate at androgen deprivation therapy, but ultimately they fail and develop castrate-resistant disease, and that's what men generally die from. So we were interested in metabolism in prostate cancer, and one of the first things that we looked at a good number of years ago was treatment of hormone-responsive prostate cancer, and these are LNCAP cells. And if you treated them with an androgen, R1881 is a synthetic androgen, you induced oxidative stress in the cells. And that's measured here by the DCF assay. So that the cells not only were responding to androgens and growing, but they were developing reactive oxygen species within the cells. And that led us to wonder whether the induction of reactive oxygen species by androgens in prostate cancer or in prostate cells contributed to prostate carcinogenesis, and if you had prostate cancer, to possibly progression of disease towards a hormone refractory state. There were others like Bostwick lab that showed that radicals such as the hydroxy radical could make changes in the DNA, and that was more present in prostate cancer tissue than normal prostate tissue. So we started to think that if androgens induced oxidative stress in the cells, how could that be? How was that occurring? How were the androgens turning on processes in metabolism that would create the oxidative stress? And at that time, we did a gene screen, much different than today, and what you could get done. But one of the genes that showed an increased expression in prostate cells with the treatment with testosterone or other androgens was SSAT, that's spermidine acetyltransferase. Prostate tissue is very high in polyamines, so these are common enzymes in prostate tissue. And what generally happens is that if you increase SSAT, you acetylate the polyamines in the prostate. That increases the substrates for other enzymes like APAO, which is acetylpolyamine oxidase. And when APAO acts on the acetylated polyamines, it creates hydrogen peroxide. So we felt that this was one of the potential sources for reactive oxygen species with androgen treatment of prostate cancer cells. We again looked and we found a small molecule that would inhibit APAO. And in fact, if we treated the cells with this small molecule, we prevented the induction of oxidative stress in the prostate cells when we added androgens to those cells. So we felt that we were onto something in terms of how was androgen inducing oxidative stress in prostate cells, and very well could have been through the metabolism of polyamines. So we also took that small molecule, which we termed APC-200, which we have a patent on now, and we treated a prostate model called TRAMP. And before we sacrificed the animals that had prostate cancer, we injected them with hydroetidine, which is a reflection, if it's taken up in cells and fluoresces, it's a reflection of oxidative stress in the cells. So not only did APC-200 reduce the number of tumors that this model developed for prostate cancer, but as you can see in the fluorescent pictures, that the cells that did form tumors had a higher rate of oxidative stress. And those were the ones that did not receive the APC-200. We thought that we would move that and look at human tissue. So with Wei Wang at the University of Wisconsin, we collected almost 400 samples of prostate tissue. And that included normal tissue, BPH tissue, PIN, low-grade cancer, metastatic cancer, aggressive cancer. And as you can see, as you go from normal to cancer to more aggressive cancer, metastatic cancer, the amount of SSAT expression in the prostate tissue goes up significantly. So again, SSAT levels seem to correlate with the degree of developing prostate cancer and the aggressiveness of prostate cancer. So then we wanted to know, well, if SSAT activation was the mechanism of creating oxidative stress, then what was the mechanism that we activated SSAT? We knew that it was necessary to add androgens, but how did the androgens ultimately affect the expression of SSAT? If you look at the promoter region of SSAT, you see a number of interesting things. What you don't see are androgen response elements. So there was nothing in the promoter region of SSAT for the androgen receptor to sit down on the DNA. There were there were sites for the glucocorticoid receptor, and there were AP1 binding sites. And as you know, AP1 consists of a cluster of proteins. And it's that cluster of proteins that can induce a gene to be expressed. So again, we screened for all the possible AP1 constituents. And the one constituent that came from the screening was an increased expression of June D in prostate cancer tissue. So of all the possible elements of AP1, June D was the one that was increased. And that, in fact, had a site in the promoter region that it could sit and turn on the production of SSAT and then go down the pathway through APAO to develop the oxidative stress. So then the question was, how would the androgens affect June D? And there are, in fact, publications from a variety of different laboratories showing that the androgen receptor can have protein-protein interactions with proteins such as June D. So AR, in fact, did not need an ARE on the promoter of SSAT to turn it on. It could perform its activity of stimulating SSAT by binding to June D. The other thing that we found, and if we were inducing oxidative stress, as you know, when cells are subjected to oxidative stress, a number of other pathways are turned on, one of which is the NF-kappa B pathway. And in fact, one of the constituents of that, P52, has been shown to associate with AR. And we saw that it went up when we added the androgens and induced oxidative stress. So we had two possibilities: P52 binding AR and turning on SSAT, or June D binding AR and turning on SSAT. So again, we looked for small molecules, and I'll show you the pathway that we chose, a small molecule. And in fact, when we developed a small molecule that would disrupt the binding of AR in June D or AR in P52, we no longer saw an increase in SSAT. We no longer saw the androgen-induced oxidative stress. In the process that we pulled out that small molecule is that we developed a Gaussian luciferase assay. And in this assay, which has generically been published by other groups, you place the coding sequence of a protein, let's say AR, in one half, and another protein that you feel that it's going to associate with in the other half, let's say June D. When those two proteins associate with each other, you get fluorescence and luminescence. If you have a molecule that disrupts the binding of those two proteins, you don't get the fluorescence. So it's an assay that's suitable for high throughput screening. And we screened almost 20,000 molecules looking for a small molecule that would disrupt AR June D binding or AR P52 binding. We pulled out a number of candidates. We ruled them out. They were not typical anti-androgens. They, in fact, disrupted the protein binding of these pairs of proteins. When we looked at the candidates that we pulled out from those two screens, in fact, it was one molecule that disrupted both AR June D and AR P52. It was exactly the same molecule. That had an IC50 of greater than micromolar range, 2, 3, 4 micromolar. And we felt that we needed to get more potent compounds. So we did a number of structure-activity relationships, and we called them previous MD Anderson. He produced one of the compounds. It was an analog of the one that we had pulled out. You can see here listed as the WP compound. In fact, when we take that compound and we look at immunoprecipitation of AR-G D or AR-P52, you can see in this slide in the two figures that the addition of the WP compound prevents the co-precipitation. It disrupts that protein-protein interaction. And if we look at prostate cancer cells, Galen-Cap androgen responsive cells, or C42 cells that are a subset of Galen-Caps that are castrate resistant. So they don't respond to androgens normally, they're resistant to anti-androgens. That in fact both of them are inhibited by this compound, and the C42 cells have an IC50 below micromolar range. So we gained about 10-fold activity with the structure-activity relationship experiments and development of the WP compound, which now we're bringing into mouse model. So that's one example of a small molecule that could disrupt various metabolic aspects in prostate cancer that potentially could be brought into the clinic, and we'll see what happens with the animal model. But if we go back to the promoter region, it's hard to ignore the fact that there are 4 GREs in the promoter region. But we generally don't associate glucocorticoid receptor with prostate cancer and treatment of prostate cancer, except for the fact for some recent data that's been coming out. And in this publication in Cell, this came out of Charles Sawyer's group at Sloan Kettering in collaboration with Chris Logadidis and Eleni Estafu at MD Anderson, who were conducting a study. So Chris and Eleni were conducting a study of taking prostate cancer patients, getting a bone marrow biopsy from them, giving them a treatment such as an anti-androgen, enzalutamide, and then a number of weeks later, repeating the bone marrow biopsy, and then subsequent down the road, if they progress, repeating it again. So they had a number of patients with serial bone marrow biopsies that showed the metastatic prostate cell. And the question that Charles Sawyer and his group were raising is what happens with glucocorticoid receptor as cells progress from being hormone sensitive to castrate-resistant prostate cancer. In fact, what you see in this, and you see that in the immunohistochemistry patterns here, that if you take the patients and divide them into good responders, meaning that they responded to the anti-androgen for more than 6 months, or poor responders, meaning they didn't respond at all, or they responded in a very brief period of time. Do you see in the poor responders, with the brown staining that you see here, that there's a lot of glucocorticoid receptor. But in the good responders, there's not very much GR. So this experiment, and you can see that the other graphs of the data that just show this in a different way, that if you take the patients, whether they be poor responders or good responders, they don't have very much GR when they started. But when you look to see who responded well or poorly, the poor responders had significantly more GR expression throughout these numbers of patients. So these experiments, and other experiments being done by Chris's group and Mark Titus and Delaney and MD Anderson, have consistently shown that in prostate cancer, as you transition, you start getting treated with androgen deprivation therapy, you do well, you start progressing, you start becoming castrate resistant. And it's in that transitional period that glucocorticoid receptor expression starts going up dramatically. And that raises the question as to whether there are compounds, there are agents that could be used at that point of disease during this GR transition. for example, GR degraders. And they are, in fact, GR degrader compounds that are coming into clinical trials. One other experiment that was done in this Mark Titus's group in MD Anderson, again looked at levels in prostate tissue of glucocorticoid receptor. And in fact, you can see the glucocorticoid receptor expression here goes up in a PDX model from a patient's bone metastases that was castrate resistant. And you see with brown stain with the high levels of GR. And in fact, what you also see is an increase in glucocorticoid levels in these patients, as witnessed here in the table with the cortisol levels. And those cortisol levels could be dropped by a variety of lyase and hydroxylase enzymes or compounds that inhibit those enzymes. And you see that with the VT compound or abiraterone, which is commonly used in prostate cancer. So we have a lot of evidence that in this transition state from androgen responsive to castrate resistant, that there are other things happening that could affect metabolism. Principally, one of them is the glucocorticoid pathway and the increase in glucocorticoid receptor. Again, another potential target for treatment of patients going through this phase of disease. So when you think about, again, getting back to the SSAT, one of the questions is, well, so what? You increase expression of SSAT. Does it affect the actual biology of the cancer cells? And we all know that for cells to metastasize, they go through a variety of different maneuvers. They have to get out of the primary tumor, they have to get into the bloodstream, they have to get back out of the bloodstream, and they have to invade a tissue and grow in it. So there are many different processes. One potential process to look at in the potential effect of increased SSAT is looking at migration of cells. And there traditionally have been assays of looking at cells transitioning or migrating through various collagen matrix, et cetera, we worked with David Beebe's group, an engineering group at Wisconsin, and Eric Bassoon and I worked with them on this stackable microfluidic device. The advantage of this is, number one, it was very small, so you didn't need a lot of cells. You could place the cells on the top. You would have a collagen matrix. You would put osteopontin and osteoblast in the lower chamber, and then after a period of time, you simply dismantled the device, and you already had slices of the matrix that you then could look at what cells migrated into the matrix and which ones migrated the furthest in the matrix. We looked at this with typical cell culture cells, the LNCAP cells, androgen responsive, and the C42 cells, androgen unresponsive cells, but a subset genetically of LNCAPs. And as you can see pictorially in the bar graph, that the C42 cells, the more aggressive castrate-resistant cells, tended to migrate more and in greater numbers towards the bone constituents, the osteoblast and the osteopontin. And in a study performed again with BB's group and Eric Basu and R1 grant that we had at the University of Wisconsin, we collected a number of patients that were undergoing prostatectomy, got cells from their primary tumor, subjected them to this invasion assay, and also stained them for SSAT. And we're in the process of following those patients to see which ones progress to more distant disease. And in fact, so far, what we've seen is that there are patients that have more aggressive cells that do migrate to a greater degree than other patient cells, and that the cells that migrate the most actually have higher levels of SSAT expression. And it's those patients with those highly migratory cells that, to a higher degree, small numbers still, but still waiting on a number of the other patients, but to a higher degree, they have progressed with metastatic disease after their primary therapy. So it does appear that it is possible not only to assay for which cells would be more aggressive in patients and do it in an in vitro setting, but those cells also have high SSAT expression. So when you look at these, do we see changes in invasive versus non-invasive cells in the metabolic sense? Now, this is the glycolytic pathway. And for the students here that are wondering, why do you really need to sit there and memorize the Krebs cycle and glycolytic pathway and oxidative phosphorylation and whatnot? That sooner or later, I guarantee you, if you're in research, you're going to get back to these pathways at some time. And you're gonna wish that you could remember what you had memorized in the first place. So you have the glycolytic pathway, and as you know, Moorberg many years ago, 100+ years ago, felt that cancer cells were dependent upon glycolysis in a greater degree than normal cells. And you can have the glycolytic pathway on the road to lactate and pyruvate. You can engage the TCA or Krebs cycle with that. But one of the critical enzymes in all of this is GAPDH, glyceraldehyde phosphate dehydrogenase. And that's an important enzyme in this pathway. Dating back years, people in fact used that as a control in their northern blots when people used to do such things. And in fact, back then in the 90s, since we were looking at prostate cells, Maureen Ripple and I looked at LNCAP cells, and in fact, androgens increase the activity of the GAPDH in these cells compared to cells that are not being treated with androgens. So there is evidence, and there's been ample evidence since then, the GAPDH is stimulated by androgens and important in the glycolytic pathways for prostate cancer cells. So then we looked at the metabolome, and this is just a simple metabolomic screen, commercial screen that we did. And we looked at the effects of androgens, R1881, versus anti-androgens, enzalutamide, in androgen-responsive cells versus castrate-resistant cells. And you see, comparing the 3 columns on the left to the 3 columns on the right, that there are dramatic differences between cells that are androgen-responsive and those that are castrate-resistant. When we look further to look at the product of the GAPDH, which will be an indication of the activity of the glycolytic pathway, we see that difference in the cells. And in fact, as we saw many years ago, if you take LNCAP cells and treat them with androgens, you see an increase in 3-phosphoglycerate. If you take the C42 cells that are androgen resistant or enzalutamide resistant, in fact, you don't see that increase. You see a decrease in it. What does that mean? Well, in other studies that we did with Lori Parker from the University of Minnesota, we were looking for androgen-induced oxidative stress and whether we were producing oxidized GAPDH. And there have been other publications that there are 2 forms of GAPDH. There's an oxidized form that does not contribute to the glycolytic pathway. In fact, associates with other proteins that are important to cell migration. So if you have more oxidized GAPDH, that would be a state where the cells would be more migratory. If you had less oxidized but more unoxidized GAPDH, the cells would be engaged more in glycolytic activity. And you see the difference here between the antigen responsive and the hormone refractory cells. That in the alpha cap cells with antigens, you see a lower level of oxidized GAPDH. They're engaged more in glycolytic activity. If you look at the C42 cells with antigen added, you have an increased amount of oxidized GAPDH, more engaged in migratory activity. So that led us further as we were looking at these cycles, TCH cycle, etc., as to what else is going on in mitochondrial metabolism. This is another important part for you in your biochemistry courses, to remember the various pathways that the mitochondria are important contributors to the energetics and metabolism of the cancer cells. TCA cycle occurs in the mitochondria, and as you know, electron transport occurs in the mitochondria to set up and maintain the proton gradient that occurs, ultimately producing ATP for the cells. So there are two pathways here, the glycolytic and TCA pathway and the oxidative phosphorylation pathway. And do these offer potential targets for treatment? Well, here, as you look at pathway and think, you have cells that are creating energy, or you could have this pathway headed in the other direction that are more engaged in biosynthesis. There are a number of important aspects here, including the activity of glutaminase. And people more and more are talking about glutamine levels as important in the microenvironment of cancer cells. Not only does it affect the rates of growth in cancer cells, but it also can affect the activity of immune active cells coming into the tissue. And in this study done by Nancy Walker at Baylor, in conjunction with Dan Frigo in our prostate group, she looked at ARV7 expressing castrate-resistant prostate cells. And what is ARV7? It's a truncated form of the androgen receptor that's constitutively on, and it's a subset of AR that has been shown clinically to occur in patients that are showing resistance to anti-androgens. So it's an important marker of resistance to treatment. And in fact, what she saw in these cells is that they tend, in the TCA cycle, to switch from bioenergetics to a more biosynthetic approach of the TCA cycle. But an important factor here is this glutaminase. And there are actually compounds now that are in the clinic that could be used to test against hormone refractory prostate cancer. This is one of the compounds. It's in clinical trials, and hopefully soon will be in clinical trials for prostate cancer. If you look at the clinical trials done so far, the plasma Cmax that's achieved in those studies is about 2 micromolar. And if you look at prostate cancer cells in a dish, the IC50 of the C42 cells is about 2 micromolar. So it does seem that if you can achieve those plasma levels, which already been shown clinically, that's in the concentration range that you need to affect castrate-resistant prostate cancer cells. And we hope to be starting a study aimed at glutamase inhibitors in the near future. Finally, if you get to the electron transport system in prostate cancer cells, are there targets here that can be used to treat patients with castrate-resistant disease? Despite the fact that Warburg felt that cells are more dependent upon glycolysis, there's more and more information that in fact the oxidative phosphorylation continues to be a very important source of energy for cancer cells, perhaps even more important for cancer stem type cells. And in fact, we have a small molecule in our drug development group, Julia Drietta, Phil Jones, that have developed a compound, brought it through preclinical testing. It is, in fact, in phase 1 testing at the current time, run by Tim Yap in our phase 1 group. So here's another target that we could consider for prostate cancer, and we do have prostate cancers going on that phase 1 study. And when we look, in the phase 1 study, patients are able to achieve steady-state 10 nanomolar range. And if you look at the effects of this compound on cells in the ditch, caps, or the C42 cells, in fact, we can achieve inhibition of these cells in the 10 nanomolar range. So again, the effects in vitro are looking at levels of the compound that we now know we can achieve clinically, and those studies are ongoing at the current time. So finally, I'm just going to show you some pictures of the process of autophagy